Coming up ahead in this episode of X Talk Spotlight.
Really key elements that have to come together here to synthesize an ADC that's safe. That's our most important thing when we're asking for regulatory submission, and that's efficacious. It needs to do the job. If we want our regulator to approve our drug, we need to prove that it's being effective in killing the target cells.
Hello, and welcome to X Talk Spotlight, illuminating insights from subject matter experts and industry thought leaders. I'm Sonia Hunt. In this episode, we're asking the question, how can broadening our knowledge in bioconjugate design and development help develop better medicines to treat oncology and beyond?
The number of U.S. Food and Drug Administration approved antibody drug conjugates, or ADCs, has grown exponentially in the last 5 years. However, there is still room for improvement in efficacy and in reducing toxic side effects. Drug developers are looking at novel formats such as pro-body drug conjugates, bispecific ADCs, and dual payload ADCs to maximize their therapeutic potential. In this X Talk Spotlight edition, I sat down with Dr. Petra Dietrich, Senior Vice President and Scientific Leader at Abzena, to discuss this important topic. Dr. Dieterich has more than 3 decades of experience working in drug development and leading projects for biotech companies in the development of active pharmaceutical ingredients for both clinical trials and commercial use. Thank you for taking the time in this Spotlight interview, Dr. Dietrich.
Yeah, thank you very much for inviting me, and I'm really excited to be here and to chat to you today.
So to start us off, when synthesizing an ADC, what different elements do you need to consider?
ADCs are complicated molecules. We all know that. They have different elements. So there's protein part, there's a small molecule, and then there's the linker part in the middle. So standard ADCs would be built on an IgG1 monoclonal antibody, and we have to really think about them that's very tumor specific. We want to be targeting receptors that are specific to tumor cells. We want our antibody to have a long half-life in circulation, and we want it really to be retained in the circulation, so it's not eliminated. Once the antibody has found the right receptor, we need it to be internalized into the cell so that the payload can do its job at doing the killing of that tumor cell. And we really need to make sure that antibody doesn't generate any unwanted immune responses. So that's the protein part. Obviously, on the other end, we have to have a very highly potent payload. We have to have a small molecule that is going to do the destruction of our tumor cell. And we need to think quite carefully about the mechanism of action. So different payloads have different mechanisms of action, but they're all highly toxic, and they need to be really highly potent. And we tend to want our payload to be hydrophobic enough. So if there is an opportunity to elucidate the bystander effect, it can diffuse into neighboring cancer cells and kill those for us as well. Of course, we have to bring those two elements together. So the linker and the conjugation chemistry is really, really important. The chemistry that we use really defines the structure of our final ADC and how the loading of that antibody to drug ratio looks. The linkers can have different properties. They can be cleavable or non-cleavable. And in the marketed ADCs that we have today, that both of those elements feature. And the linker chemistry really also determines the physiochemical properties. They have to be stable in circulation. We don't want to get any shedding of that payload, because that can have some quite toxic effects and also affect our potency. So yeah, really three really key elements that have to come together here to synthesize an ADC that's safe. That's our most important thing when we're asking for regulatory submission, and that's efficacious. It needs to do the job. if we want our regulator to approve our drug, we need to prove that it's being effective in killing the target cells that we're targeting.
And what are some key shortcomings that drug developers working in the ADC space have identified?
Shortcomings are really related to drug resistance and adverse effects. Traditionally, we've thought of ADCs as widening the therapeutic index. So we're looking at increasing the maximum tolerated dose and going down in the minimum effective dose. In reality, clinical trials have shown us indeed that ADCs are very effective. If we take Enhertu, Enhertu is the poster child amongst ADCs right now. It's got pan-tumor potential, and it's really been extremely successful in combating breast cancer, metastatic breast cancer. Response rates in clinical trials are up to 75%. But even there, we're seeing some significant issues with interstitial lung disease in about 9% of patients. And, disappointingly, the treatment has to be disrupted for those patients. So there's more we can do to stop these adverse effects and really make sure that we keep those cancer treatments going. So drug resistance is really the root of this, and it's brought about by tumor heterogeneity, and the different responses to treatment are brought about by the different strains in the cancer having different responses to the ADCs. And really that leads to resistant strains, and then treatment stops working, very sadly. And really that's a key problem. Adverse events are really when the toxicities become unacceptable, and treatment has to be discontinued. And really that's then alternative treatments have to be found or discontinued completely. Mylotarg, for instance, was approved for leukemia in the year 2000, but it led to a far too high number of early deaths and had to be discontinued. Since then, more data has been found. We've established different treatment regimens, and under a lower dose, we can now are authorized to reuse that. That's been since 2017, and under that dose, it's been reinstated as a treatment. We've spoken about Enhertu as well. So there's really a lot more that we can do, and really what we're looking at is to change the formats and further elaborate on what we've got already to make sure that we retain that efficacy and to make sure our drugs are safe.
Now, what solutions are being considered to overcome these shortcomings?
When we target a cancer cell, what we want to be doing is looking for a receptor that's unique to that cell, and obviously less expressed in a healthy cell, so that the healthy cells stay healthy. However, very often those receptors aren't unique to the cancer cells. They do appear in other cells. And so when that happens, you can get the ADC payload entering the healthy cells, and then you get what we call this on-target off-tumor effect, which can lead to unfortunately, destruction of those healthy cells, and then some quite devastating effects. And what people are really looking at now is something that we call probodies. So in the ADC, the AGG1 antibody molecule would contain a masking unit. So we would mask the antigen binding site, or we could also alter the conformation so that we don't get binding straight away to those receptors. And then it's only in the tumor microenvironment where we're really getting that binding to happen. And so we can be much more specific and exclusive to the cancer cells. One company that's made some great strides in this area is CytomX Therapeutics. They've made these pro-body drug conjugates, PDCs, and they've really been able to have some great results. They've increased the therapeutic index by about 10-fold in their preclinical monkey studies. And they've advanced now into clinical trials to see whether we can just have a much more effective and safe drug moving forward. There's other things we can do. They're not so advanced. We can look at bispecific antibodies. So in a bispecific antibody, you would target maybe 2 different receptors. So if you can identify 2 receptors on your cancer cell, you could maybe more exclusively bind in that region, and then obviously internalize and your toxic payload and get the cell destruction that way. There is some risk with that. You can get agonistic effects, so that you're not actually having the right targeting, but that is something that people are looking at. Another option that people are looking at is to have dual payloads. So instead of just having one single type of payload, we can have 2 different payloads. And those payloads would be chosen because they have different mechanisms of action. So the different tumors, the heterogeneous tumors, the ADC with a dual payload could target all those different types of cancer cell and then get destruction. Again, it's early days for this technology. We have to be careful in looking at those mechanisms of action and looking carefully at the underlying biology that's going on there. But there's a lot of activity in this field. The industry has certainly come a long way, and its understanding of the chemistries involved. And I think we're more than capable of synthesizing these molecules. What we have to be sure, I guess, is in the design, so that we're really designing the best possible molecule to have those desired effects.
And to wrap up, can any of the learnings from ADC development be applied to other therapeutic moieties, such as antibody oligoconjugates?
Obviously, over the years, we've built a plethora of techniques for conjugating the different targeting proteins with a range of different payloads. So we can have a standard monoclonal antibody with a high potent auristatin MMAE. That's a favorite payload at the time. And we can vary that and apply lots of different design options. We can take that another step. So we can take a targeting protein like an antibody fragment, and we can couple it with an oligonucleotide. So really exciting, quite a different molecule, completely different mode of action here for treatments, but we're still using the chemistry that we've learned from our ADC synthesis to apply to this new molecule, just using the different elements that are available in our toolbox. So oligonucleotides really impact the protein production. So they would interfere with the expression of the protein. They could destroy it, they can modify, or they can enhance. So if we have diseases that are caused through proteins, the oligonucleotide therapies can really impact at that level of the protein expression. So there's different types of oligonucleotides. There's antisense oligonucleotides, small interfering RNA nucleotides, and these can all be synthesized now using specialist chemical techniques. They can be stabilized, and we can really start using them to alter the protein expressions and to really start having an impact on diseases that are caused by these elements. Oligos aren't easy to handle. They're large hydrophobic molecules, 4,000 to 20,000 kilodaltons, negatively charged, really have limited bioavailability. And we've done some horrible things such as directly injecting them into eyes and injecting them into spinal cords, really want to be looking at a more systemic delivery method, something that's a little bit easier to handle. And really bioconjugation has really offered itself as the way forward here. So we can go back to our ADC synthesis and we can choose very similar linkers. And if we use a bifunctional linker, we can link one end to the protein using the standard techniques. So through cysteine or lysine conjugation to maleimides on one end of the function. And then we would probably choose maybe an azide to link through to the oligo to make that connection between the azide, between the oligo and the protein. So really just using techniques that we already know, but applying them to different moieties. The other thing that I think we've learned from ADC molecules is that we can modify the linkers. We spoke a little bit about hydrophobicity, can be a good thing, can be a bad thing as well. So on the ADC side, we have the option to put something like a PEG molecule or sometimes a carbohydrate like a glucuronamide molecule to enhance that hydrophilicity. And that's an option that we use in ADC development very frequently. We can apply that to oligos as well. We can modify our linkers. We can add units. We can add PEG. Obviously, the hydrophobicity isn't so much of a problem, but there are studies that show that if we add PEG, we can increase the half-life in circulation, which is something that we always want to do. So, just going back to those chemistries and really learning the connections that we've made and applying them in a different situation has really shown us the way to make these molecules, to make them more easily administered for patients.
Well, thank you very much, Dr. Dieterich, for speaking with us today. We really appreciate your time and insights.
It's been an absolute pleasure to be here today and to talk to you a little bit about ADCs and other conjugates.
We look forward to learning more about Abzena's work in the antibody drug conjugate space. Thank you all for joining us for this X Talk Spotlight feature. We hope you enjoyed the discussion.